Thank you, Ernest. Thank you for the invitation to speak. It's really good to be here. I've not been to PAG before. I'm an evolutionary genomicist. For those who don't know me, I spent 25 years in Edinburgh being an academic, teaching a lot of undergraduates genomics and doing genomics on the side, as it were. And a job came up at the Wellcome Sanger Institute to help run the Tree of Life program there, which has just started. And it's my dream job. I've always wanted to do something like this. I spent my PhD sequencing at the rate of 1,000 bases a year. That was last millennium. And now I'm at a place where we hope to be doing, this year alone, we hope to be doing 1,000 chromosomally resolved genomes. So I'm at the Wellcome Trust Sanger Institute. You may know the Wellcome Sanger Institute historically as a place that did human genomics. And that's true. The Sanger Institute has been involved in human genomics for 26 years now. But Sanger Institute started by sequencing not a human, but sequencing Cenorhabditis elegans, a small free-living nematode. It was the first animal genome sequenced. And actually, the Sanger Institute, through the decades, has sequenced many things apart from humans. So zebrafish, mice, thousands upon thousands of bacteria, and lots of parasites and pathogens and their vectors. So the Sanger Institute, if you like, has been doing a low-intensity, all-biota genome sequencing project for the past 25 years. But spurred by many people, but especially by this paper, which some Sanger Institute authors are on, came the idea of participating in a really crazy project, which is to sequence all eukaryotic life on Earth. So this Earth Biogenome project, proposed by Harris Lewin and others, the idea is to generate high-quality reference genomes for every single species on Earth. That's somewhere between 1.5 and 2 million known species. Why do this? Well, to me, it's because we know from, well, the C. elegans genome project since 1998, but also the Human Genome Project, that once you have a reference genome, all sorts of biology are possible. And the human genome sequence has transformed the way we do human biology. And what we'd like to do is transform the way that all biology is done. I know you all buy into that idea because you're all working on your own species, and you know what change having that genome will affect. And so what we want to do is do that for everybody's science. There's lots of reasons why you might want to sequence everything rather than just a few representatives. These are the arguments given in the Earth Biogenome Project paper in PNAS, and ones that we at the Sanger Institute join up to. We'll understand how life evolved. We'll understand the processes of genome evolution. We'll understand the rules that life has evolved under. And we'll also understand how our biosphere is responding to climate change. We're in the middle of the 6th great extinction. It behoves us to really think and look at that. In the same way, these genomes would be great tools for use in conservation and management, and also in thinking about how to do conservation, not at the level of individual species, however important those individual species are, but the level of whole biota, all the organisms in a region. And as we move to a post-oil economy, as we move to an economy which must reduce its reliance on fossil fuels, these genomes will provide the raw materials, the raw sequence materials for this economy. So new genes, new gene systems, new genetic systems to make new biologicals, whether they're feedstocks for materials or whether they're feedstocks for biopharmaceuticals. So on the basis of that, we're going to sequence everything on the planet. We don't own the planet. I think that's really important to understand that at the Sanger Institute, we're part of the British Isles, Britain and Ireland, and we have a rather sad colonial history of having made a mess of the rest of the world in Britain. So what we decided to do is sequence our own biota first. So that's not just a parochialism thinking we're going to sequence our own backyard, but really because by looking in our backyard, we can actually look at the whole world. There's also issues to do with, well, major issues to do with the Nagoya protocols and the Convention of Biological Diversity, which means we shouldn't sample elsewhere without permission. So we've started something we call the Darwin Tree of Life Project, which is the Sanger Institute plus partners sequencing all the organisms in this small archipelago of islands just off the west coast of Europe. We call it the Darwin Project, partly because everything you call Darwin suddenly has extra kudos, but also because we want to do the same thing as Darwin did. So Darwin did go around the world. He did get inspiration from all the biology around the world. When he came back to Britain, he never really left home again. He went, he got a placement in Down in Kent, and lived in that house in Down in Kent for the rest of his life. Corresponded with people around the world, not by email, but real written letters, and did experiments in his garden. And he understood the whole world, the whole of evolution, from his back garden, not just the origin of species, but also all his other books right up to the formation of vegetable mold through the action of earthworms. That's based on experiments in his garden. And so these animals, plants, fungi, and protists that live on this little archipelago off the west coast of Europe are actually one of the best known biotas in the world. They've been studied for centuries. They have an engaged community of naturalists and scientists working on them with very detailed records. And so we're going to start here as an example for the rest of the world as to how a genome project covering a whole biota can happen. The Sanger Institute isn't a biodiversity institute, despite having sequenced quite a few different species in its history. And so we're collaborating with biodiversity partners throughout the UK and Ireland, working with the Natural History Museum and the Botanical Gardens. They embody all of what we know about species. We're also working with specific sites, and I'll talk a bit about Wytham Woods near Oxford, but also with Marine Biological Association, who are interested in the marine realm. We're also working with biodiversity partners who are interested in genomics. So we're working with the University of Cambridge on assembly technologies, with Earlham Institute, who are very interested in the assembly of single-celled eukaryotes, and with EMBL, EBI, who are going to be hosting all these data and making it public. So what we're going to do, well, what we have in Britain is a list of species. So this is the list. It's, despite all those hundreds of years of looking, it's still not actually quite complete. There are still species we know must be missing from this list, and there's still argument about what some of the species are. But there are about 60,000 species described on these islands. So next to the total global diversity of 1.5 million species, that's a tiny proportion, so 0.04%. 0.04. But what we have is a diversity in terms of families. So if you look at the families in Britain, there are over 4,000 families of eukaryotes. That's plants, animals, fungi, and protists in the UK and Ireland. And using those, we actually cover about 40% of all the diversity at the family level across the planet. So by sequencing the species in Britain, we really do get to a lot of the biodiversity of the world. So our goal we've set ourselves is to sequence to near chromosomal completeness all the species in Britain and Ireland. And we're going to start with getting reference sequences for all the families. Okay, so only 4,000 genomes. Organizing to that is quite difficult, so we started in a slightly more scattershot way, but we are aiming to do 4,000 families in the next 3 years. After that, we'll move on to the genera, and after that, we'll do the rest. This requires us ramping up by, in about 5 years' time, doing 5,000 genomes a year. If you divide that by the number of working days in a year, that's 20 genomes a day. That's 5 before coffee. Another 5 before. It's terrifying. But I think actually we can get there. I'm standing up here representing a large group of people, not only the biodiversity and analysis partners, but a large group of people at Sanger Institute who really believe this is possible. So what we're going to do this is find the species in the wild, identify them, make sure we've got the right species, and what we're going to do is DNA barcode those individuals so we have a marker, a record. We then have to extract long DNA from those species, do our sequencing, whatever method we choose, do the assembly, and release those data publicly. And so we're building a pipeline, a factory, an engine that we can put a snail in the top and get a genome at the bottom. Efficiently and without having to go back and look at each one individually. So how are we going to do this? We're going to use two kinds of data. Long read data, and it's really important that it's the change in the quality and the length of the long reads that makes this at all possible. 2 years ago, this would have been a hilarious idea. Now it's just scary, because I think we can do it. Those long-read data can come from Pacific Biosciences, especially the hi-fi data to us appears very useful, but also from Oxford Nanopore Promethean. The other type of data we're using is long-range data. So long-range means data which not necessarily sequence, but tells you that 2 bits of sequence are on the same linkage group on the same molecule. And the best data for that in our hands so far is Hi-C, the chromosome capture data, but also 10x data, the link-read clouds from 10x, and also bio-nano restriction fragment maps are really good for putting chromosomes together. We're going to sequence from individual specimens of each species, and that's really important to us. Our experience of sequencing, putting 50 moths of the same species in a blender and trying to sequence that, is that we're trying to sequence more than 2 haplotypes at any one time. We're trying to sequence something which really is very complex, and so the assembly graphs that we get out don't resolve, and the assemblies are really quite poor. So we're going to minimize haplotype confusion by sequencing from individuals, and we're also going to try and get to fully phased assemblies. We're not there yet, but we hope with the data types we're getting that that's going to be possible. So we're actually going to get both haplotypes assembled. And that's really important if you want to use these data for all sorts of downstream applications. But chromosomal assemblies is what we're aiming for. So I'm going to give you an example of where we are based on the White & Woods site. White & Woods is a lovely old forest, or bit of woodland, near Oxford. It's been studied as an environmental and ecological research site for about 80 years. There's lots of data about not only the abiotic factors like temperature and so on, but also biotic factors. There's a good species list. And it's now being developed as a genomic observatory, a place where we can observe the evolution of genomes in real time. And so the White & Woods team has been collecting lots of samples and sending them to us. So we have a lot of samples in the freezer already, so from about 221 species. And in terms of our goal of sequencing families, that's about 100 families. That number will go up hugely later this year. As spring happens and life comes back to White & Woods, there'll be many, many species. One of the things that's been very easy to collect over the autumn was butterflies and moths. So we have about 80 species of butterflies and moths in our collection ready to sequence. So we've been doing that. And this is really where it shows the power of the hi-fi data. So this graph has, along the x-axis, the length of the assembly we have achieved with the hi-fi data. When I say we, I mean Shane McCarthy and his team. We have achieved with these hi-fi data just straight out of the box. So we're not doing anything about haplotype reduction here. So these moth assemblies, butterfly moth assemblies, range from about 280 megabases up to 1.4 gigabases. And the red dots in this graph are the assemblies we've generated. The y-axis is the contig N50. This is the contiguous nucleotide N50 of these genomes. And they're all pretty good. So all the ones on this graph are above half a megabase, and most of them, except for 3, are above a megabase contig N50. The blue dots in this graph are some of them are my previous life. Before I joined Sanger, I was involved in several butterfly genome projects, and we worked very hard, sometimes for years, to generate genome assemblies which had brilliant N50s, none of which were significantly very much above 100 kilobases. So all the blue dots is previous life. The red dots are the butterflies we sequenced in the last, butterflies and moths we sequenced in the last 3 months. The pink line is the average chromosome size you get if your genome had 31 chromosomes. So interestingly, most butterflies and moths have 31 chromosomes. We don't know that's true for all these species. But that means that already we have assemblies with N50 contig sizes above the expected chromosome size. There's also the expected relation to genome size. As the genomes get bigger, our contigs get smaller. There's also a relationship with coverage. There's also a relationship with repetitive content. That's not shown here. What's really interesting is when we put the hi-fi data with the Hi-C data. Haven't done that for many genomes yet, but all of these are ending up chromosomal. So this is telomere to telomere assemblies of these butterflies. When we check how good they are, it looks like they're really good. So it looks like they are collinear with what we expect. So we think we can get to, for these genomes, which is up to about 1.5 gigabases, we can get chromosomal assemblies out of 2 data types. And as I say, having spent years sequencing other butterflies, this is truly transformational. We hope we can spread this across the whole of the tree of life. So this is a wonderfully colorful and maybe too complicated graph of where we are. The Sanger, as I said, has been involved in other projects at the same time as the tree of life, been involved in the Vertebrate Genomes Project, also sequencing other vertebrates associated with the Vertebrate Genomes Project. This is where we are. About 158 species went through the Sanger Institute and generated a significant amount of data last year. And inside the Darwin Tree of Life, so this is the UK sequencing project, there are 94 official Darwin species which have generated data, assemblies, or have been submitted to public databases. So I just want to quickly comment about that submitted bit. What we're doing with these data is releasing them immediately to the public databases. So this morning, our first genome note was published. This is a 1-page paper. It's in Wellcome Open Research, which is based on the F1000 platform. It's a 1-page paper saying we have sequenced the genome of the red squirrel, Sciurus vulgaris. Here it is. And at the end of the paper, it says, this genome is now free for people to use. So we're not putting any embargo on these genomes. These genomes are going to be published as fast as we can. So we hope to build a process that takes the snail, makes the DNA, generates the sequence, generates the assembly, and publishes the paper, the genome note, seamlessly so that these data are available for all. So this will hopefully release the floodgates of people using these data for post-genomic research. It's not to say there aren't problems. There are problems in finding everything in Britain. There are 60-odd thousand species described. How do we find them all? There's a wonderful species which is 8 feet long, which you think would be quite difficult to lose. It's called the bramble shark. If you don't know about the bramble shark, look it up. It's a glorious shark. Sharks have denticles on their skin. The bramble shark has great big spines on its skin, like the spines on blackberries, on brambles. It's been seen in British waters twice. Once in 1870 and once in 1970. Both on the North Sea, so it's relatively limited. So we could spend a couple of £1 million dredging the North Sea for the next few years and catch one more and sequence it. I don't think anybody would thank us for that. I think we might just wait and see if somebody else finds a bramble shark somewhere else. They're all around the Atlantic. So there are some things which are so rare we may never find them. There are some species which may not actually exist, either because they've gone extinct or because they were mistakes in the first place. But our main problem is actually going to be the species that are small. So most of life is tiny. Even most of animal life is tiny. The myofauna and the little no-see-um gnats that bite you when you're on the beach. So getting to those organisms and sequencing their genomes is going to be really hard. And we've got no doubts that that's going to be hard. So we're working hard to try and get to that. At the moment, we're at about half a mosquito is the amount of DNA we want. And this paper was published a year ago now, giving a very high, showing you can get a very high quality de novo genome from half a mosquito using PacBio sequencing. So in this case, the genome that came out compared to the one that was sequenced using Sanger dideoxy sequencing has a much, much better contig N50 and is much, much better in terms of completeness and contiguity than the old one. And so we can go down to half a mosquito. The next job is going down to something that's even smaller, the individual tiny organisms. So that's a big challenge we've got. In the first 2 or 3 years, we're mostly going to be sequencing bigger things. But soon we're going to have to challenge ourselves with these tinies. OK, so I think it can be done. I don't think I would have taken the job if I didn't believe it could be done. But I think we now have, in the first 6 months or so, data that proves that we can actually sequence all the species in the British Isles. What's going to be really exciting is moving on and working with others to sequence all the species on the planet. So just say it's a collaboration. It's not just us at Sanger. It's also the people in the biodiversity partners and in our analysis partners. And even at the Sanger, it's not just the people at Tree of Life, Project, who are the ones in green here, but also the people in the Sanger Scientific Operations who are working really closely with us to get these technologies working, and also people like Jason and colleagues at PacBio who are helping us get the best out of their technologies. So there's a lot of names on this list, but your name could be on this list too. Of course, I'm allowed to put up a job advert. We've got a set of jobs open at the moment for a project we're initiating in symbiosis genomics through the Moore Foundation. So we're sequencing about 1,000 symbiotic pairs, that's 2,000 genomes in the next 3 years with the Moore Foundation project. That's lab people and bioinformaticians we're looking for. We're looking for people to collaborate with us on analysis of these data. So this is people who have established positions elsewhere who might want to work with us and spend some time at Sanger working on key projects. And we also have additional posts in databasing, LIMS, and informatics. If you're interested, either talk to me or Shane and Kirsten, who are sitting here. And the last thing to say, because I'm 23 seconds over, is we've got some stickers for the Darwin Tree of Life logo. So if you want to take home a sticker to remind yourself that we're doing something quite as crazy as this, please do. Thank you.